Just wait for a moment. Very good evening. I hope you can hear and also see the screen. We'll just take few questions today and then we'll stop. Tomorrow is your exam, so all the best for that. I hope you all can hear me and also see the screen. Can you please just let me know that? The screen is visible and I am audible. Oh, yes. Thank you so much. So, without wasting much time, thank you, Sakshi. Thank you, Raj. So, without wasting much time now, we'll directly go to our. Yes, Priyanka. Yes, Tej, Tajal. Great. So, we have the first question here. All of you already know me. I'm Professor Girish Kukreja, and we are preparing for your entrance examination. There is an entrance examination tomorrow, and like I was like कि ये लेना है या वो लेना है ये लेना है तो थोड़े मिक्स कर दिए क्वेश्चन तो थोड़े से जस्ट एक क्वेश्चन हम ओवरव्यू करेंगे आई नो इतने इजी क्वेश्चंस नहीं आने वाले बट आई डिडंट हैव अ गुड प्रेजेंटेशन रेडी मे बी if you are continuing for the next week I can make some good presentation where we may have more lengthy questions being discussed so these are only one word type of easy questions only to revive your brain cells कि थोड़ा एक्टिव हो जाए थोड़ा आपको कुछ याद आ जाए तो यही कल आएंगे ऐसा कुछ नहीं but then still we can have an overview of this right. So yes, here you have a complete beta oxidation of a 16 carbon fatty acid. That day only you told that a 16 carbon fatty acid was a palmitic acid, right? Which was a saturated one. What will it produce? A complete beta oxidation, right? You remember those 4 steps of beta oxidation? I'm not discussing them. The NADH, the FADH2, 2 NADH it says. Only one fatty acid and only 2 NADH. Oh my God, no. 4 FADH2, 8 molecules of acetyl-CoA and this complete beta oxidation of a 16 carbon fatty acid requires 8 cycles for its completion. So which thing you think is true for a complete beta oxidation of fatty acid? It gives 2 NADH, it gives 4 FADH2, it gives 8 acetyl-CoA or it requires 8 cycles for its completion. Yes, which one you think is true? Ross, Mahajan, Priyanka, Tajal, Sakshi, which one you think is true? Yes, please. I'm waiting for your answers here. Yes. A complete beta oxidation. I'm sure it is not going to give 2 NADH, it is not going to give FADH. Dozens of them are produced. You have like more than 120 ATPs being produced from one 16 carbon fatty acid. So surely it is not going to be this 2 and 4. Now you have a crux of this 8 acetyl-CoA and 8 cycles. Like what do you think? Yes, great, Ross. It is 8 acetyl-CoA. It even doesn't require 8 cycles because it will require 7 turns, which will produce what you call as remove the 14 carbons, and the last one which is there, the eighth one, it will be like directly acetyl CoA, right? You know that fatty acid doesn't enter into the cell directly, right? Fatty acid, not cell actually, like in the mitochondria where it's being oxidized. Fatty acid is first activated to form fatty acyl CoA, and you know that there is a transporter which carries this what you call as fatty acyl CoA into the mitochondria. Can you name that small molecule which is actually carrying this, shuttling this fatty acid from cytoplasm into the mitochondria? What is that small molecule called as? No, yeah, it requires 7 cycles. Great. So that small molecule, you remember though that small molecule, the arginine or the fascicine or the acetine or the carnitine. What do you think is the molecule? Anyone, if you remember, which is that molecule which does this? ACP. Oh, yeah, that acyl carrier protein, Ross, is involved in the synthesis of fatty acid. I'm saying when I'm going to take this fatty acyl CoA into the mitochondria, which small molecule is going to help me? I gave you the options: the arginine, the acetine, the histidine, or the carnitine. No, Priyanka, it's not acetine. I'm so sorry, it's not acetine. It is. I'm not wasting much time because I know that you're too sleepy, and so am I. So it is carnitine. Remember, it is the carnitine molecule which actually helps in transporting your fatty acid from what you call a cytoplasm into the mitochondria. I would just like to know that when the fatty acyl CoA, which is being formed in the cytoplasm, is being transported into the mitochondria, it is the fatty acyl CoA which goes in, or it is only the fatty acid leaving back the CoA? What do you think? वो CoA अपने साथ लेके जाता है या सिर्फ fatty acid अंदर जाता है और अंदर फिर CoA से दूसरे मिलता है? What do you think? It takes the same or it takes different? Atharva, what do you think? Atharva, it's not arginine. I'm so sorry. It's a basic amino acid. It's carnitine. Okay, better luck next time. I want to know कि जो fatty acid अंदर जा रहा है जब mitochondria में, तो I know that it's converted first into fatty acyl CoA in the cytoplasm itself. So I just want to know that CoA, it carries itself with inside the cytoplasm or inside the mitochondria, or it leaves that CoA in the cytoplasm, goes with the carnitine and takes a new CoA from the mitochondria. So I would like, you can just comment the same CoA or it leaves that CoA. Atharva says same. I'm again so sorry, Atharva. It's not the same CoA. It leaves the CoA there itself. क्योंकि हर बार वो फैटी एसिड जब अंदर आएगा उसको वो CoA अंदर लेके जाना पड़ेगा और फिर वो CoA बाहर लाना पड़ेगा. तो सेल क्या करता है? It keeps that CoA outside. The cytoplasm wala CoA is kept in the cytoplasm, and then mitochondria there is a separate coenzyme A which is going to what you call as transacetylase this fatty acid which has come. So fatty acyl CoA combines with carnitine, leaves the CoA, it enters inside, combines with another CoA, so it doesn't carry the same CoA, remember. Anyways, different. Yes, Ross, it is different. Let's go to the next one. During this degradation of triacyl glycerol, you know, a triacyl glycerol contains one glycerol molecule and 3 fatty acids, right? A triacyl glycerol contains what? One fatty acid and no, not one fatty acid, one glycerol and 3 fatty acids. When it is being degraded, fatty acids will go in beta oxidation. What about the glycerol? The glycerol, it also undergoes beta oxidation, or it goes in the pathway of glycolysis, or it enters the citric acid cycle, or it forms ketone bodies. Yes, it's a different CoA. Yes, I would like to know this. ये glycerol का क्या हुआ? Triacyl glycerol में से आपने glycerol निकाल दिया, fatty acids अलग निकाल दिए, glycerol अलग निकाल दिया. Yes, Ross, it undergoes in glycolysis where it can be converted to your dihydroxyacetone phosphate or glyceraldehyde 3-phosphate, from where it can like continue its pathway. So glycerol, it enters glycolysis. Great. It enters glycolysis. which of the following is made of only one type of macromolecule? सिर्फ एक ही टाइप के मैक्रोमोलिक्यूल से इसमें से क्या बना हुआ है? वायरस, प्लाज्मिड, राइबोसोम या न्यूक्लियोसोम, which contains only one what you call as molecule? Yes, great. It is DHAP glycerol, as you said, the 3-carbon one. One type of macromolecule is present in what? A virus, a plasmid, a ribosome, or a nucleosome. What do you think? One type of macromolecule. Virus, plasmid, ribosome, or nucleosome. I know my board is small. Handwriting is not good. It is not focusing properly, but I hope you can still do that. Yes, great. Atharva says plasmid. Wonderful. Yes, Atharva, you're right. It is the plasmid because it is made only of the DNA, right? Virus will have DNA or RNA or protein. Similarly, ribosomes, is made of RNA and proteins. Nucleosome also of DNA and proteins, right? Anyways, nitrogen fixation with azola in the rice. It has which organism with it? It has spirulina, it has anabina, it has frankia, or it has rhizobia. No, Ross, it's not ribosome because ribosome has both, right? It has RNA as well as protein. So it has two types of macromolecules, right? Ribosomes are ribonucleoprotein particles. You remember who observed ribosomes for the first time? Yes, Atharva, it's plasmid. Yes, it's anabina. Great. It is a blue-green algae or cyanobacteria. So it is your anabina. Spirulina, we know that's associated with your single-cell protein. Rhizobium with the leguminous plant. Frankia, can anyone guide me what frankia is? Yes, anabina is right. Can anyone guide what frankia is? It's a Frankie, which my wife makes sometimes for dinner. No. What is frankia? Can you just tell me what is frankia? It is a bacteria or a fungus, at least you can tell that. Frankia is a bacteria or fungus, at least that much. If you could have you heard about frankia? I hope you have. Frankia is, it's not fungus, Atharva. I'm so sorry. Anyone else now? Obviously, if it is not fungus, it is going to be bacteria. Can you tell me that frankia, does it help in nitrogen fixation? Yes or no? You can just type Y or N. Yeah, great. It is a nitrogen fixing actinomyces. Is a wonderful answer, Ross. Yes, it's a bacteria. It's a bacteria. Actinomyces itself is a bacteria. Anyways, it helps in nitrogen fixation or not? Y or N? Yes, great. It does that. The numbers here 36, 34, 36. Oh, sorry. 36, 34, 30, 20, 24. Oh, this seems to be so easy. Oh, I got it. Can you tell me the next one? 22. Great, great. Atharva is good in competitive exams. So, right, this is -2, this is -4, this is -2, this is -4, this is -2. Great, Atharva. I hope you did it right. Great, great, great. Yes, Raj. I don't know. Y'all are more good at mathematics than biology. Don't change the stream now, but we really need some good mathematics in biology, right? Solubility of protein is least at its pi, right? Pi, the isoelectric point, is the point where the charge on the protein is 0. So at that particular point, the protein is least soluble. So what do you think? True or false? Prachi says yes, yes. I don't know. She's saying yes for that cyanobacteria or she's saying yes for this. There you can write T or F. True, great. So it is least soluble, right? Because it is not having any charge. So when it is not having any charge, so similar charges, they repel each other and therefore they are in the solution. But if they are not having any charge, then they can come together and they will precipitate. So they are least soluble at their pi. So I think that you can also be like that when we are not carrying our egos, and us like I am carrying my own ego, that the samor wala person is also carrying his ego, she's also carrying her ego, and then when you come together you cannot be together, you have to be separate because your egos are keeping you separate. But then now when you're not having a charge on yourself, when you are neutral, right, when you have no charge and the other person is also having no charge, then you can come together, then you can be one. Oh, great message. Great. True. That's true is for this, I know, not for what I said, right? The non-competitive inhibition can be overcome by increasing the substrate concentration. What do you think? The non-competitive inhibition can be overcome by increasing the substrate concentration. True or false? A non-competitive inhibition you can overcome by increasing the substrate concentration. मतलब एक नॉन कॉम्पिटिटिव इनहिबिटर है, आप सबस्ट्रेट डालते जा रहे तो एक पॉइंट ऐसा आएगा कि देयर इज नो इनहिबिशन एंड देयर इज नो चेंज इन द Vmax. Will it happen with the non-competitive inhibitor? A non-competitive inhibitor, you remember that it has nothing to do with the binding of the substrate. So whether the substrate is bound or not bound, your non-competitive inhibitor is going to bound your enzyme. So when it has nothing to do with the substrate, obviously increasing the concentration of substrate is not going to do any good to it. Maybe a competitive inhibition you can overcome by adding a lot of substrate, because now when there is a lot of substrate, the enzyme can bind either the substrate or the inhibitor, because the binding site for the substrate and the inhibitor is the same. तो आपके सामने दो चीजें दोनों सेम दिख रही है, तो जो ज्यादा है आप उधर ही जाएंगे, जो कम है वहां पे नहीं जाएंगे आप. तो competitive inhibition you can overcome by adding more substrate. But a non-competitive inhibition, right, where the binding site of the inhibitor and the substrate is completely different on the surface of the enzyme, it cannot be overcome by increasing the concentration of the substrate. So this is false. This is true, as you said. Great, Atharva. Let's go to the next. Elimination of the plasmid is called as. What is the elimination of plasmid called as? Like when you remove the plasmid from the cell, what it is called as? It is called as curing, breaking, fixing, or expulsion. You can just comment A, B, C, D. Curing, fixing, breaking, or expulsion. A, B, C, D. I hope you can see the board. Curing, wonderful. So you go for plasmid curing many of the times in your experiments. The unit of specific activity of the enzyme. Great. No, it's not D, it's not expulsion, you're not expelling it. Unit of specific activity of the enzyme, when you're calculating the specific activity, you calculate it as the units of the enzyme upon milligrams of your protein. So what is the unit here? Units per ml, units milligram, units per milligram, or units per milligram per ml? What is it? Yes, please. Specific activity. As I told, specific activity is units of enzyme upon milligram of protein. तो अभी तो simple कर दिया मैंने. Yes, U per mg. Yes, great. It is units per milligram. Can you tell me, like, as the purity it increases, or as the purification steps are going forward, with the increasing the number of steps, or if you really think that we are on the right direction, this happens only when the specific activity increases, decreases, or remains constant? What to expect about the specific activity when you are purifying the enzyme? After every step, the specific activity should increase, should decrease, or should remain constant? Yeah, great. It should increase. Okay. So here we got this. Which aspect is not altered by the enzyme? Like you are carrying out a particular reaction and you're adding an enzyme in one case and you're not adding the enzyme in the another case, right? So what will not change whether you have added the enzyme or not added the enzyme, whether the rate of reaction will not change, whether the thermodynamics of the reaction will not change, or whether the activation energy will not change, or whether the time for the reaction will not change. What will not change? You can just comment as A, B, C, or D. What do you really think does not change? The rate of the reaction, the thermodynamics, the activation energy, or the time for the reaction? B, great, Atharva, great. The thermodynamics, it does not change, right? Because the free energy of the reaction, right, before and after, the initial and the final stage are the same. The time, the rate, and the activation energy all decrease. Rate increases actually, the time decreases. Great. B, great, great. Thermodynamics, great. The non-translated sequence in the transcript at the start of the translation site. आपने translation शुरू किया है, but शुरुआत में there is a region which is there in the transcript but is not being translated. What is that? It is the ending frame, it is the leader sequence, it is the reading frame, or the trailer sequence. You can just comment as A, B, C, D. I read it again. The ending frame, the leader sequence, the reading frame, or the trailer sequence. Great, great. Atharva says that it is the leader sequence. Great, great. It is the leader sequence. Great. The first bacterial genome to be sequenced when this human genome projects and all were going on that time, they thought कि चलो लगे हाथ bacteria का भी sequence करते. So which particular bacteria was the chosen one? The Bacillus subtilis, the Haemophilus influenzae, the E. coli, or Methylophilus methylotrophus? C, it's not E. coli, I'm afraid. It's not E. coli, Prachi. No, Priyanka, it's not E. coli, it's not C. It's not C, Atharva, it's not C. E. coli, आपका favorite है, but no, it was not E. coli, Ross. B seems to be better. Thank you, Meghna. B seems to be Haemophilus influenzae. Yes, that is the one. The nonsense codon, the one which doesn't have sense, is your UAA, UAG, UGA, or all of these. A very, very basic question, I suppose. The nonsense codon. We all know this. Great, great. It is all the nonsense codons are all of these. Great. We are done with those questions actually. I have a few more questions here with me, so I hope you can answer. I hope I'm audible now. I'm not writing it on the board. It's not B, Prachi, it's D, all of these. So I hope I'm audible and you can see me and you can hear me, right? So I'll just be asking question and then you can just give the answer, right? Okay, great, Prathamesh, it's all. The type 1 hypersensitivity, right? When we are talking about hypersensitivity, there are different types of hypersensitivity. You know who gave that classification? You know that classification of hypersensitivity? Anyways, if you could tell me the name who gave those classification in the comment, that will help you to recall and others to recall that. Anyways, so type 1 hypersensitivity, can anyone just comment, is caused by gel and Coombs? Great, Vivek, wonderful. Your immunology seems to be good. The type 1 hypersensitivity, as I was talking, is caused by which antibody? IgM, IgD, IgE, IgG. The type 1. Type 1 is not delayed type of hypersensitivity, right? It is not DTH. Type 1 is IgE. Great, great. Type 1 is IgE type of antibody. Great. And you know that this IgE goes and eating and wandering minds. Oh, nice to meet you, eating and wandering minds. Wonderful names you have, right? I really love them. I also like to eat and I also like to wander. So anyways, I'll talk to you later. Do you know coagulase? The enzyme coagulase. It is produced by which bacterium? Can you guide me? Coagulase is yes allergen specific IgE beta. Yes. Coagulase. Can you tell me it is produced by whom? It is produced by Streptococcus epidermidis, Streptococcus saprophytis, S aureus. Great. It is S aureus. Great. Gram positive bacteria, they don't contain something. Right? Can you just guess what gram positive bacteria does not contain? The teichoic acid, the plasma membrane, the outer membrane, or the murine? What does gram positive bacteria does not contain out of these? Endotoxin, yeah. So that is the outer membrane which acts as the endotoxin. If you could just tell me this. No, I'm not asking. This is very easy. Antigen antibody complexes will involve formation of all types of bonds except the hydrogen bond, the electrostatic bond, the hydrophobic bond, or the covalent bond. No, Prathamesh, it's not teichoic acid. It is what others said. It is the outer membrane. Gram-positive bacteria do not contain outer membrane. Teichoic acid is rather a specific character of this D. She says covalent bond. Great, great. Covalent bond. Yes, covalent bond is not there. You know that some bacteria are resistant to some antibiotics like penicillin. So great, great. It's covalent bond. I would like to know who is this eating and wandering minds. If you could tell me the name and the city, that would be so nice. So I could come there to wander. Bacteria resistant to penicillin and some related antibiotics there. They produce an enzyme. Can you know? Can you tell which enzyme it is? Right, the bacteria. Oh, hi Rutuja. And you're from where, Rutuja? If I could know. Yes, Karthik. This is which Karthik? Karthik from where? Anyways, you go with it. The bacteria which are resistant to penicillin, right? What do you think that which enzyme they produce? Oh, great. Thank you. Beta lactamase. Great. It is your beta lactamase. Great. I have many more. But do you know this TATAAT box? So this box is of about 10 bases, which is present 10 bases before the start point. Yes, penicillinase is beta lactamase is a better answer, I suppose, because it leaves the beta lactam ring. Anyways, the same one. TATAT is present 10 bases before the start point. What it is called as? The start box, the row box, the Pibno box, or the enhancer box. What it is called? The start box, the row box, the Pibno box, or the enhancer box. If you could tell me the name, I really forgot the sequence. Tata box or the Pibno box. Yeah, that is better. Thank you so much, Meghna. Meghna, you're from where, if I could know? Yes, Atharva. Yes, it is Pibno box. Great. Your genetics seems to be good. The study of genetics, obviously, I mean. The Line-Weaver and Burke plot, right? You all know the Line-Weaver and Burke plot, the LB plot. It is a straight line graph which is obtained by doing what to your Michaelis-Menten equation? You multiply it by V0, or you just take a reciprocal of all the terms in the Michaelis-Menten equation, or you multiply everything by S, or you are dividing everything by V max. What do? You're multiplying it by something, or you are just taking an inverse. Yeah, you are just taking an inverse. Great, great, great. I'm not taking many of them. I'm itself tired, as I told. The Ames test, you know the Ames test. So Ames test is done for what? Yeah, reciprocal. Yeah. The Ames test is done for what? Right? For can you tell me for what the Ames test is done? AMES, Ames test. Or let me modify. Let me make it easy for you. Carcinogenic mutagens. Great, great, great. For mutagen screening, and you use a specific auxotroph for it. Can you tell like which auxotroph is used in this AIM test? Yeah, carcinogenicity testing or mutagenicity testing it is used. But can you tell like which auxotroph is used in this? And you study the reversion of that auxotroph, right, in this AIMS test. If you could tell which auxotroph is used. Yes, please. Yeah, carcinogenicity. But which auxotroph? I mean, like that S. typhimurium is auxotrophic for what? Do you really know that? Like it is auxotrophic for what? Yeah, S typhi strain. Meghna, I would like to know from where you are. Yes, S typhi. I am sorry for my question. I wanted to ask, like, it is auxotrophic for what? Just a moment. Histamine. Histamine. I really don't think so. Histamine. मैं रॉन्ग हो रहा हूँ या आप रॉन्ग हो रहे हैं? Histidine, now there you are. Leptospirosis, right? Leptospirosis, you all know leptospirosis. Now, this leptospirosis is caused by whom? It's caused by chlamydia. I'll be specific, so you can answer as A, B, C, D. I'll be slow. Leptospirosis is caused by A, chlamydia, B, rickettsia, C, protozoa, or D, spirochetes. Leptospirosis is caused by A, chlamydia, B, rickettsia, C, protozoa, and D, spirochete. Now you can be more specific, like A, B, C, D. Only 4 to go, not even till Z. Spirochetes, great, Rituja, it's spirochetes. The Shine-Dalgarno sequence, it's not A, Priyanka, it's D, the spirochetes. The Shine-Dalgarno sequence, you know the Shine-Dalgarno sequence, it's a part of eukaryotic mRNA, prokaryotic mRNA, eukaryotic rRNA, or eukaryotic tRNA. The Shine-Dalgarno sequence. Where will you find the Shine-Dalgarno sequence? The eukaryotic mRNA, the pro-mRNA, uRNA, or uTRNA. You can just write D, I don't know, like this is for what? I forgot the sequence. I forgot the sequence. You just tell me it's like pro or U, and it can be like PM or ETEREM. Yeah, prokaryotic. Yes, Omkar. Great. Thank you so much. Thank you. It is prokaryotic mRNA. The protein which helps to fold other proteins in a proper manner and prevents the misfolding of the protein. Now I am being specific, Omkar. The protein which helps to fold other proteins and prevents the misfolding of that particular protein is A. I'll be specific, as I said, I'll be specific. A, ubiquitin, B, proteasome, chaperone, right? So desmosome and chaperones. Thank you, Meghna. Yes, it is chaperones. So Meghna is like reducing my work of giving options. The lac repressor, you all know the lac repressor, right? The sequence from which this lac repressor is being produced, you can label that as the operator or the promoter. A, operator, B, promoter, C, inducer, and 4, regulator. Can you tell me? A, operator, B, promoter, C, inducer, and 4, regulator. What can I be more specific about? Yes, chaperones is right. What can be more specific about that gene which is coding for the repressor? Operator, promoter, inducer, or regulator? Yeah, great, Atharva, great. It is regulator. Wonderful. The molecule, it's not promoter, it's not operator, it is the regulator, right? Because the repressor will bind the operator. I am saying the gene which is coding for the repressor. Yes, Pratmesh, it is the regulator gene. Great. Yes, Ross, it is the regulator gene. A few more, 2-3 more only, right? So then we can have a good night. 1 or 2 more only. I've skipped many anyways. the bacterial quorum sensing, right? So the bacterial quorum sensing, when it is taking place, the molecules which are involved in this bacterial quorum sensing are A, amino acids, I'll be specific, A, amino acids, B, flavonoids, C, catecholamines, and D, acetylhomoserine. A, amino acid, B, flavonoids, C, catecholamines, and D, acetylhomoserine. Which molecule do you think really is involved in the bacterial? It's not catecholamines, Ross. I'm so sorry. It's not catecholamines at that extent, at least. D, Sakshi says it's D. Prachi also says acetylhomoserine. Meghna, yes, great. When I say that an organism is oxidase positive, right? When I say that the organism is oxidase positive, I'm actually looking for which enzyme? The hydrogen peroxidase A, the catalase, the lyase, or cytochrome oxidase. Like for which enzyme I'm looking for? Hydrogen peroxidase, catalase, lyase, and cytochrome oxidase. D, great. It is the cytochrome oxidase. Great. Great. Cytochrome oxidase. Wonderful. The acid fast staining, like for what purpose you use phenol? It's not catalase, I'm so sorry. Catalase nahi hai. That is cytochrome oxidase. For catalase, you will do catalase test. Why do you do oxidase test, right? Acid fast staining, right? You know, acid fast staining. What is the role of the phenol there? It acts as a fixative, it acts as a mordant, it acts as a solubilizing agent, or it acts as accentuator. What is the role of phenol in acid fast staining? Yes, cytochrome oxidase is the right answer, Prathamesh. What is the role of phenol in acid fast staining? Fixative, mordant, solubilizer, or accentuator? Accentuator. Great. Last one here. The del factor indicates the kinetics of the death of microorganisms by dry heat, moist heat, desiccation, or radiation. Dry heat, moist heat, desiccation, or radiation. The del factor. Dry heat, moist heat, desiccation, or radiation. The last one. Wonderful. It's the moist heat. Great. And all the best to all of you. I hope you are like very well prepared for tomorrow's exam. And if at all you wish me to continue, yes, Ross, it's moist heat. Great. If you think that we should continue with more such sessions, you want me explanation, want from me actually the explanation of some topics, so you can ping me and we'll see whether it's possible. So stay tuned with me, your friend for biology, and keep sharing, keep loving, and really all the best for your tomorrow's examination, right? So God bless you all, and really good wishes from my side, Professor Girish Kukreja. Thank you and good night, and best wishes for tomorrow. Maybe we can connect tomorrow evening to discuss how your exam was, and was this really helpful or not, right? And there are some exams which are actually scheduled later, maybe in the next week, and if you think that we can meet every day for 10-15 minutes at least, and that will really help you, let's try to do that if you really think so. And as I tell in my every session, my number is 9823683317. It's 9823683317. So you can ping me so that we can be connected. And thank you so much. Bye bye. If you have anything else to say, you can just comment here, so we can be connected. Miss Ingray, you are also preparing for MSc entrances. Meghna, I would like to know, like Meghna is from here. Yes, Vivek. Thank you. Vivek, your birthday treat is still pending. After tomorrow's exam, you can call me at least for a cup of tea. We can join for a cup of tea at least. I'll be waiting. Great. So best wishes to you, Tuja. I would like to know Meghna is from where, and there was Omkar, someone who had joined, Karthik. Oh, great, great, Meghna. Okay, all the best and thank you so much. Good night. Bye bye. Take care. Yes, Vivek, I'll be really waiting. Good night. Yes, Atharva, I know. We just got connected. You're from Jalgaon. If others could please tell their state or the city, that will be really helpful to know from where you are connected. Thank you. Yes. Good night, Atharva. Bye bye. Anyways, we'll meet tomorrow, then we can discuss. Bye bye. I hope so. Onkar, please connect to me on WhatsApp. And may I know you are from where? Pune. Great. I would like to connect to you. You can ping me on my WhatsApp. I've already shared my number 9823683317. Bye bye. Thank you. My son is waiting for me to sleep now. So I'm really going now. Bye bye. Good night. Take care.